The genetic basis and fitness consequences of sperm midpiece size in deer mice.
The researchers examined two species of deer mice, Paramyscus maniculatus and Paramyscus polionotus. Paramyscus maniculatus is known to be promiscuous, meaning that it is capable of mating with multiple partners, and Paramyscus polionotus tends to mate with only one partner, meaning it's monogamous. The purpose of this experiment was to test whether male mice's sperm mid-piece length is correlated with the swimming performance and whether longer piece length leads to higher reproductive success.
The midpiece, also known as the mid-connecting piece, contains dense fibers and mitochondria for the sperm. When the researchers conducted a statistical t-test comparing the means of the length of the sperm midpiece in the two species, the midpiece region in the promiscuous species was significantly longer than the monogamous species. The size of the midpiece was also predicted to positively influence flagellar thrust and sperm velocity, increasing the likelihood of achieving fertilization.
As we have learned, DNA is transcribed to RNA, which is then translated into protein. In the Paramyscus mice, the researchers discovered the PRKAR1A gene, which encodes the R1 alpha regulatory protein, a subunit of protein kinase. R1 alpha is the protein that is known to affect the sperm morphology and motility. Even though these proteins were found in similar amounts in the mice's tests, they were expressed differently in the two species.
To correlate the size of the midpiece length to the motility of the sperm, the researchers conducted swim-up assays. First, when the sperms of the sister species, the promiscuous species and the monogamous species, when they were raced, the most motile sperm was found to have a longer midpiece. And even when the sperms of the two independent promiscuous species and within the same promiscuous species were examined, the longer midpiece variations were more motile, which showed that longer midpiece suggested a higher success of fertilization.
Previous studies have shown that sperm competition can be driven by the number of sperm from rivaling males, but this study demonstrated that the quality of the sperm can also influence who will sire offspring. Fisher and Hoekstra, in another study, showed that sperm competition between the promiscuous species is driven by another adaptation called the cooperative behavior. The sperm of the competing promiscuous species have the ability to recognize each other and aggregate, which can increase the sperm velocity. Those that can form these aggregates can have a selective advantage upon competition.
When P. maniculatus and P. polionotus were intercrossed, 300 F2 males were produced. When these were examined, it was discovered that these male hybrids with at least one allele from the promiscuous species at the particular gene locus associated with morphology and motility had a longer midpiece than the male hybrids with none. The figure above represents the sperm performance and the male fertility in the F2 hybrids. In other words, it compared the midpiece length to the straight line velocity of the sperm for both species. The VSL is correlated with midpiece length either by an increase of midpiece length leads to an increased speed, or the two traits are independent of each other by tightly linked genes. The P. maniculatus has a longer sperm midpiece than the P. polionotus. When these two species compete with each other in the experiment, P. maniculatus had a selective advantage over this other species because of the longer sperm midpiece. This also shows that the male P. maniculatus has a stronger reproductive success over the other species.
In conclusion, the researchers claimed that the promiscuous species produces sperm with a longer midpiece than the monogamous species, and midpiece size correlates positively with competitive ability and swimming performance. They ultimately showed that their hypothesis was supported because, in this case, sexual selection favored sperms that had a longer midpiece. They also showed that those with a longer midpiece were also more likely to reproduce, which suggests that in nature it may be possible that the deer mice will eventually have the longer sperm midpiece adaptation.